This video will explain how to set up a simple microecosystem to investigate food web interactions. In this experiment, the brine shrimp Artemia salina will be our herbivore. Our producer will be from the genus Tetraselmis, which is also known as Platymonas, the genus of green algae. Here is a list of the materials you'll need for this experiment. You'll need a spectrophotometer, seawater, algae, a micropipetter, graduated cylinder, jars, a transfer pipette and petri dishes, and a dissecting microscope. First, we will measure the concentration of our algae. If you're unsure about these methods, please refer to the video of how to use a spectrophotometer. Pipette 2 milliliters of seawater into your cuvette to use to blank the spectrophotometer. Next, you're going to determine the concentration of your algae stock. Using a clean pipette tip, transfer 2 milliliters of your algae solution into a clean cuvette. Place the cuvette in the spectrophotometer and record the value of the absorbance. Using the worksheet provided, calculate the algae concentration of your sample using the standard curve. Next, label your jars. On each lid, place a piece of tape and label it with the group name, the day, the time of your class, as well as the treatment that will be inside that jar. Next, you're going to add the calculated amount of seawater to each jar. You'll use your worksheet for calculations of how much seawater to use to dilute your algae culture. Measure out the required volume of seawater, remembering to read from the meniscus in your graduated cylinder. In this case, we've calculated that we'll need 45 milliliters of seawater and 5 milliliters of algae. So we first add 45 milliliters of seawater to the jar. Now we'll add the appropriate amount of algae to our jar. Now in this example, we'll need to add 5 milliliters. So first we swirl the algae to mix it. Then we select 1 milliliter using our micropipetter with a clean tip. Add that to the jar, and then repeat 4 more times. Next, you're going to add the brine shrimp to each jar. First, collect a sample from your brine shrimp. You see here that there is a mixture of swimming, moving brine shrimp, as well as some unhatched eggs. Those are the small yellow spheres. You're going to collect some of that sample, transfer it to a petri dish, and then look at the sample using a dissecting microscope. This is an unhatched egg. You don't want to select these to add to your ecosystem. Instead, select the brine shrimp like you see here. Carefully use the pipette to select individual brine shrimp to transfer to your ecosystem jar. You may need to dilute the brine shrimp culture using additional seawater so that you're able to pick up just one individual. The focus of this experiment is to investigate trophic interactions. Let's look into that further. There are 2 types of interactions between trophic levels. Let's imagine an ecosystem that's made up of plants, insects, and birds. In this ecosystem, the insects eat the plants and the birds eat our insects. Now in a bottom-up interaction between trophic levels, as the number of plants increases, so does the number of insects. And as we increase the number of insects, we see a higher number of birds. And so if we look at the number of plants, as that increases, the number of birds increases too. Now in a top-down interaction, as the number of birds increases, we'll find fewer insects. And as the number of insects increases, there will be fewer plants. And so if we look at the relationship between birds and plants, as the number of birds increases, the number of plants also increases. In a bottom-up, if there are more plants, there are more insects, and there are more birds. But in a top-down interaction, if there are more birds, we see fewer insects and more plants. As you plan your experiment, think about what you would need to manipulate to study bottom-up and top-down interactions using algae brine shrimp microecosystems.